The overall goal of this method is to generate representative shock waves that can occur during blast events and evaluate the impact of this mechanical shock on the viability of primary cells in culture.
This method can help us answer key questions relating to the response of individual cell types to high-energy events, such as exposure to a shockwave. The main advantage of this method is it enables us to evaluate distinct cell responses to a single shockwave as opposed to tissue or organ-level responses.
The implications of this technique extend towards identifying the cellular mechanisms altered as a result of primary blast injuries, which are caused by exposure to a shockwave.
Though this method is being demonstrated with a specific cell type of interest to our laboratory, any cells can be cultured and placed within the test setup to assess cell-specific responses to a shockwave.
To begin, culture the cell line of interest at standard culture until they reach 80% confluence. Then, aspirate the medium, wash the cells twice with PBS, and add 2 milliliters of trypsin EDTA. Incubate them at standard culture conditions for 5 minutes. Briefly check to ensure the cells have successfully dissociated from the flask, and then add 4 milliliters of FBS-containing culture medium to the flask in order to neutralize the trypsin and transfer the cells to a 15 milliliter conical tube. Centrifuge the cells at 200 times G for 4 minutes to pellet the cells. Then, slowly aspirate the supernatant, taking care not to dislodge the pellet, and resuspend the pellet in 5 milliliters of medium.